Well, welcome everyone. Welcome to Straight Science. Straight Science is an evening seminar series put on by University of Alaska Fairbanks Northwest Campus here in Nome and University of Alaska Fairbanks Alaska Sea Grant right here. And tonight you're in the home office. And both UAF Alaska Sea Grant and Northwest Campus. We are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. So tonight we have Leah Valdes, and she has just completed her PhD in entomology at Cornell University. But as you can see, and Jay, I might ask you to just turn your camera off just for the bandwidth, but we'll let
Okay,
Leah and Haley on. Thank you so much.
you're welcome.
And we have, so Leah tonight, so back in a few years ago, Leah and Haley Schofield, and we all know in Nome, Haley Schofield, she is the Climate Change Program Director at COERIC. And both Leah and Haley have done work in the region on bees. And I have to say, we do straight science with huge research vessels on the moose. And you hear these people come in and talk about walrus and the climate, the world at large. And yet there has been so much interest. Both times bees have been discussed. So we're really excited to have Leah and Haley come back and talk to us about their research on the bees. And it looks like, Leah, you are up at bat. We're so grateful for you are here, and tell us about our busy summer neighbors. Thank you so much. Take it away.
Oh man. Awesome. Thanks so much, everyone, for coming. I am psyched to talk to all of y'all today about what Haley and I have found, what the Alaskan bumblebees have taught Haley and I about really how bumblebees defend themselves against disease. And so like Gay just said, this was all done. I thought I was going to give a little introduction on Haley because I didn't know she would be joining us today. But all of this was like a close collaboration between me and Haley Schofield, who is also a awesome honeybee biologist. So yeah, everything I'm going to talk about, it's kind of a team project. Haley just tapped me to be the one to talk about it today. Yeah, so with that, I wanted to begin by talking generally about infectious disease. So infectious disease is a global threat to the health of both managed and wild animal populations. Just to give a few examples of this, white nose syndrome arrived in the US in the 2000s, and it affects hibernating bats. And it's led to little brown bat declines of up to 90% in some places in the US. Chytrid fungus has been decimating amphibian populations globally. And then y'all probably, I don't need to tell y'all about the snow crab fishery, which has been affected by bitter crab disease, which leads to unpalatable flesh and population declines that have significant economic impacts. And then the avian flu, I think we've all experienced the price of eggs right now, and it's crazy. So understanding how to effectively manage infectious disease in both managed and wildlife populations represents a significant goal, not only of wildlife conservation, but also of food security. And I think bees really well represent a species that exists on both sides of the spectrum. Like we need wild native bees to pollinate plants in wild ecosystems, as well as subsistence plants like blueberries. And we also need domestic bees to do the agricultural pollination to produce the food that we eat. So Alaska is home to many, many different pollinating insect species. Just to give a few examples, this includes mosquitoes, surprisingly, solitary bees, flies. So this looks like a bumblebee, but it's actually a bumblebee mimic fly. And you can kind of tell because it has those like big bulbous eyes and these little tiny antennae, as well as moths, just to name a few examples. But I'm going to be talking about bumblebees today. And bumblebees represent especially significant pollinating insects in Alaska, and generally just in high latitude and alpine ecosystems globally. And this is because they have, oh, I just clapped. This is because they have adaptations that help them deal with cold climates. They have these big, fluffy bodies with like big, robust flight muscles. And this allows them to fly when it's cold, sometimes when snow is still on the ground, when it's windy, and even when it's raining. And I'm living in Juneau right now, and we've just had the second wettest April that's been documented. And I still see bumblebees flying around and like dodging those raindrops. And you can imagine in Southeast Alaska, not a lot of pollination would happen if we didn't have bees that could handle those kinds of wet conditions. They also have the ability to pollinate some significant plants like blueberries, because they have the ability to do a special kind of pollination called buzz pollination. So bumblebees, unlike most bees, can like really shake the plant in a way that causes pollen to come out. And most bee species, honeybees, can't do this kind of pollination. So yeah, we really need bumblebees in the pollination of blueberry specifically. Yeah, Alaska is also home to 22 different bumblebee species, which is quite a lot. That represents half of all species in the whole United States. Um, and so I also wanted to just point out when I'm talking about bumblebees, I'm talking about a whole complex of like 22 species, not just one. And they all have totally different lifestyles and preferences and fill different niches. But y'all have probably seen the apocalyptic media coverage about bee declines. And indeed, this isn't just type. Across the US and Europe, bee declines have been documented across many different bee species. And this includes range contractions and declines in abundance. And all of this has the potential to have significant negative effects, both in wild ecosystems and also in the food system. And what is causing these declines? So it really seems like it's no one thing. And the common consensus right now is that these bee declines have been driven by a combination of pesticides and fungicides. And this kind of makes sense. You know, if you put insecticides out and the bees ingest those insecticides, they might not do so well as insects. Habitat loss. So this includes both loss of nesting habitat and food availability, and is especially challenging in places with agricultural intensification. So if you have a huge field of alfalfa, for example, that's not going to provide great nutrition to bees who, much like humans, really do best with a varied diet with lots of different plant species. And then you guessed it, I'm going to be focusing on infectious disease, so parasites and pathogens. And I want you to keep in mind throughout this talk that all of these causes interact to affect the changes in bee populations, even while I focus in on pathogens. Yeah, but although the status of bumblebees has been extensively studied in the contiguous US and Europe, we know much less about the status in Alaska and much of Canada, as well as what causes may be contributing to those changes. And this is just because long-run monitoring is relatively limited in Canada and Alaska, although this is definitely changing, which is really exciting, and I will talk about that in a few slides. But so we might predict different things to be going on in the bumblebees in Alaska opposed to, say, the Midwest. Not only do different species live in these places, but they face different conditions. So in the Midwest, bees might be dealing with agricultural intensification and things like pesticide use and monocrops and habitat loss. While in Alaska, which has much less ag, they might be dealing with challenges associated with climate change and maybe changes in delicate alpine ecosystems. But without additional data, it's pretty difficult to draw these conclusions about bee populations. So the whole goal initially in this project was just to collect some field observations of Alaskan bumblebees and the pathogens that they host in West Alaska. So Haley and I did this along the Nome road system. And this site selection was really just that Haley grew up in Nome and it was logistically feasible and hadn't been well studied in the past. But this was really, driven by logistics and Haley's connections. Yeah, cool. So we focused on the following aims. One, what bumblebees live in this area. Two, do gut pathogens infect these bees? And then three, if so, how do these bees defend themselves against the pathogens? But initially, I'm just going to focus on aim one. So in this section of the project, Haley and I drove transects along the road system. This map is showing one of those transects. And we stopped at sites separated by about 5 to 10 miles each. At these sites, we walked transects on both sides of the road and used hand netting to collect bees that we saw on plants. We then collected these bees and pinned them and labeled them. And then all of these bees ended up being contributed to a big project way beyond Haley and me. So we sent them off to the Alaska Bee Atlas Project and the UAA insect collection. And this is a big collaborative project that's being led by folks at UAA to fill in those gaps across the whole state of Alaska and document bees, which is really amazing. And it was awesome to be able to just help them out and contribute some bees. If you want to get involved, again, I'm not directly involved in this project, except in that Haley and I contributed bees to them. But you can check out this link to the Alaska Bee Atlas website. I think they especially would appreciate, they've been using citizen science to fill in some of these gaps in bee distribution. And I think they'd especially appreciate samples in these red tiles, which have relatively low past sampling. So if you want to get involved, you should totally check out the cool stuff they've been doing and follow this link. So once we sent off the bees, Jessica Reichen and Leaf helped us. Well, they ID'd all of the bees. And so this was 360 bees, just so many bees, representing 11 species once all the identification was said and done. Which, to remind you, Alaska has 22 bumblebee species. So this is about half of all of the species in Alaska, which is pretty amazing, just along the Nome road system. Yeah, really amazing range of species. Through the next section of the talk, I'll just be focusing in on Bombus frigidus, because this was the most abundant species that Haley and I found in the sampling. And so it was the one that we wanted to focus on in the pathogen side of the study. So the next step was to address if gut pathogens infect these bees. So this is a taxa that I studied in my East Coast bees in Ithaca. So this is Crithidia, which is a genus of gut pathogens. It has a global distribution, so we thought it would be quite likely that we would find it in the bees in Nome as well. It's been found in Europe, Asia, across most of the contiguous US. And it's been found to have negative effects on bumblebees with things like colony establishment. So infected queens don't effectively establish colonies as often, longevity, and so infected bees have an increased chance of dying, and cognition. Sometimes infected bees have trouble figuring out how to manage plants. And these negative effects can be especially negative in bees challenged by something else. So like a bee that hasn't had enough to eat. Like I said, it's a gut pathogen, so it kind of lines the bee's intestine and reproduces inside the bee's intestine, and then is transmitted when those bees poop it out someplace and it's ingested by an additional bee. And as you can see in this little photo on the left, bees can be quite poopy, so this is an issue. I also included this video of a microscope slide of some bee guts. So I took the guts out of the bee, I squished them out, slapped them on a slide, and that's what this video is, to show you what these pathogens look like swimming in bee guts. So I'm going to play that real quick. You can see I'm kind of swimming around. Play it a second. The video is being second. Oh, cool. And Crithidia species have been detected in bumblebees in Canada and in West Alaska, like around Delta Junction. So we thought it was quite likely that we would detect them in Nome bees as well. But we wanted to test this just to, you know, be confident. So we took a sample of Bombus frigidus bees, which we collected. So this was 50 bees, and we chose Bombus frigidus because it was the most abundant species, and we thought we wouldn't have too much of a risk of negatively affecting a local bee population. And we inspected it via light microscopy. So dissected the gut and put the gut on a slide and looked at it in the microscope and just counted the pathogen cells that we saw in that sample. That was my extremely scientific field setup to do these dissections, and a video of one of the Nome bees' guts. So it has all kinds of wildlife swimming around inside of it. I don't know what this thing on the top section is, but we definitely identified Crithidia species in many of these bees' guts. So yes, we found Crithidia infection in 29 out of the 50 bees screened. So this is a prevalence of about 58% infected, which sounds like quite a lot of infection. If 60% of people had a disease, I would not feel good about that. But this isn't too exceptional. So estimates of Crithidia infection across the rest of the US show a ton of variation, and we don't really know why that is and what causes different levels of C. bombi infection. But it can range from no bees infected to up to 82% of bees infected, which is huge. And so the 58% estimate is definitely on the high end of the range of infections, but it's not too unexpected. So yes, we found that gut pathogens do infect these bees at a prevalence of about 58%. But bees don't have to be defenseless against pathogens. And next we wanted to test address how the bees defend themselves against those pathogens. So to talk about that, I kind of need to begin by talking a little about pathogen transmission in bee communities. So bees can become infected with pathogens when they visit an infected plant and ingest that pathogen. So maybe they eat some pollen and the pathogen was in that pollen, and then they fly off. And if it establishes inside the bee, it can then spread that infection to an additional plant when it visits a different plant and defecates on it. And so the cycle continues. And the ways in which the bee community and the plant community affects pathogen transmission in bees has been relatively well studied in the last few decades. So that's things like, how do the shape of plants affect pathogen transmission? How does the abundance of bees affect pathogen transmission? But one big gap is missing, and that's what happens when the bumblebees go home at night. So you probably know bumblebees have a social lifestyle. They live in colonies of up to 200 bees, up to 200, it depends on the species, up to 200, 250, 300 bees. And they live in those colonies in small enclosed spaces, which could lead to rapid within colony pathogen transmission. But this stage has been relatively unstudied up until now. And so, like I just said, you might imagine a colony of a bunch of bees living in a small space might experience a lot of within colony transmission. And we really need to understand, if we want to understand disease in bee communities, I think we really need to know what's going on within those colonies. But this has been challenged by the fact that it's usually prohibitively difficult to locate bumblebee nests in temperate meadows in the field. Bumblebees nest below ground, often in abandoned mouse holes, and this can make it really difficult to find them. And then on top of that, these meadows that they nest in often have dense vegetation. And if you look out at this meadow, I think you can imagine it might be difficult to see a bee flying into a hole in the ground. Um, to help you visualize that, this is a photo of the entrance of one bumblebee colony, and it just looks like a pile of sticks. So it's really difficult to find these colonies to study within colony transmission directly, at least in the Ithaca area, in which I was doing a lot of my projects. But Haley and I found something much different in the Nome area, and I don't really know why this is, but when we walked transects to collect bees in the census stage of the study, we found sometimes we'd be walking along and then we would step on a clump of moss and it would buzz at us. And in that way, we ended up finding a bunch of bumblebee colonies. So it's possible that the abundance of bumblebee colonies is just exceptionally high in this region, making it relatively easy to locate them. And it could also be that the vegetation is being a little less dense, makes it less difficult. But I really don't know what the cause was. What I do know is that we found 10 wild Bombus frigidus nests in the field. That's Haley digging one up. And so this gave us this unprecedented opportunity to address this question in the field of within colony pathogen transmission in bumblebees. So this slide is showing on the left panel what these bees look like. This is just a representative sample of Bombus frigidus. And you can see it's a really cute, fluffy bee and quite small. And then in the middle panel, two habitat types, typical habitats in which we found these nests. So residential lawn on the top and a tundra ecosystem on the bottom. And then in the final panel, what these colonies look like. So Bombus frigidus has relatively small colonies of up to like 20 bees in the nest that we found. And you can kind of see this in this last panel as well. But what I really want to emphasize, and the coolest finding of all with these nests was that we found that when we investigated the nests, all of them had this main nest cavity that was nice and clean and had the eggs, the bees, the food provisions, and then a cavity next to it that was cold and wet and filled with poop. And when we took a sample of 3 of these colonies out of the field and brought them into the lab, we found that the bees continued to defecate in 1 to 2 concentrated locations, kind of on the edge of the nest, but outside of that main nest cavity. And we thought this was a really exciting observation of defecation hygiene. We called this outside cavity, the poop cavity, the outhouse. And we hypothesized that this hygiene could be preventing the transmission of disease within these colonies. So addressing aim 3, how do bees defend the nests from these pathogens? We found evidence that they may be pooping outside the nest, but we didn't link that directly to the pathogens in these field observations. To do that, we really had to move into the lab to test directly if the outhouse protects against disease. We needed to do some manipulative lab experiments that we couldn't really do with these wild bees. So I want to take a little bit of a step back and talk about disease a little bit. So disease is often thought of as a cost of social life by behavioral ecologists. And this makes intuitive sense. As colony sizes increase, the frequency and duration of social contacts between group mates also increase. which can be really favorable to pathogen transmission. And indeed, across avian and mammal species, we often see that pathogen prevalence and intensity increase with group size, especially in contagious and directly transmitted pathogens. But social species can also cooperate to reduce disease. And we saw this in the COVID-19 pandemic through things like social distancing, masking, and the vaccine initiative. But animals do this too. And when animals do this, we call it social immunity. So social immunity encompasses all of the ways that animals cooperate to avoid, control, and eliminate infections. So in honeybees, that includes things like using antimicrobial resins in the nest, allo grooming, so like cleaning off a nest mate, and waste removal. And I'm going to be focusing on waste removal because this is a fecally transmitted pathogen, and because we also had those field observations of what looked like waste removal in the Nome bees. So waste removal is a widespread is widespread across taxa. We see it across mammals, insects, humans, and it's generally thought of to have a social immunity function to like prevent disease. And in humans, we even take this to the next level and bake it into the built environment. So human homes usually have some kind of bathroom, outhouse space to provide spatial separation between us and contagious materials such as poop. And so Haley and I hypothesized that that's what the bees in Nome had been doing as well. So they have this separate bathroom outhouse space within the nest, which allowed them to remove waste into that bathroom space. And this slowed and we hypothesized that this slowed pathogen transmission and increased fitness. Now, we couldn't just kidnap the Alaskan bees and bring them all into the lab. That would have been bad. So we had to pivot to a lab species. This is Bombus impatiens. And so this is the most common bumblebee species on the East Coast, and it's kind of the lab rat of bumblebees. It's really easy to breed in captivity. Not really easy, but easy enough to breed in captivity. It has these robust colonies that just make it really amenable to lab study and these kinds of manipulative experiments. So in this study, we addressed the following questions. One, do bumblebees modify the nest by excavating outhouses in which they isolate feces? So do they actively like dig out these spaces? Two, does the removal of feces prevent within colony C-bomb transmission? So does it actually, does this waste removal actually have a social immunity function? Three, finally, does this outhouse actually, is the outhouse itself enough to slow C-bomb transmission and improve colony growth and fitness? So I'm just going to begin with question one. So to test if bumblebees actively modify the nest, we set up some mini colonies in the lab. So all of these colonies had 25 bees in each, and I set them up in cages in which half of the cage was filled with this soft, like foam material, and half of it was open. And the bees could kind of manipulate and dig in this foam if they wanted to. We also fed the bees green food dye to make it really easy to see the feces. And we gave them about a week just to live in the lab and see what they did. And we found that the bees, most of the colonies dug out cavities and tunnels in this foam block. So they did manipulate the nest, which is really cool. So you can see this one has this kind of cavity dug out on the side of the nest. And when we look at the defecation, we also found that in the colonies that modified the foam block, most of the poop was concentrated on this half of the nest that was beneath the foam and beneath the outhouse, and the nest was relatively clean. We did have one colony that didn't manipulate the foam. Don't know why it chose not to, but 7 out of 8 of them did. And in this last colony that didn't manipulate the foam, they obviously defecated within the nest, but kind of on the edge of the nest, like in this final panel. When I quantified the amount of poop in each location, this is what I found. So on the y-axis, you have the amount of poop in each location, in the nest in gold and the foam side in blue. And you can see we found significantly less poop in the gold nest cavities than in the blue foam cavities. Yeah, indicating that the bees defecated on the side that they manipulated. But so in that last experiment, the bees didn't have access to an outside space. They just had access to the nest cage. And so we wanted to test if the bees would continue to defecate in this outhouse cavity, even when they had access to like an outside space. So to test this, we set up mini colonies again in the lab. So they had a main nest cavity, a connected outhouse cavity, and then a big flight cage that they could fly around in that had pollen and sucrose and natural like daylight, sunlight conditions. Of these, we inoculated half of them with a sham control and half of them with the pathogen C. bombi. We also fed them all red food dye this time to make the feces really easy to see. And qualitatively, we saw, so all of the pink represents the poop. We fed them red food dye. And you can see, obviously, the outhouse has the most feces and the densest concentration of feces, while the feeding location and nest have much less. Again, quantitatively, we see this kind of trend across all 18 of the colonies that we tested. So this is showing the amount of poop in each location on the y-axis, in the feeding location, nest, and outhouse, respectively, on the x-axis. And you can see the outhouse has the most poop, and this wasn't affected by infection status at all. So yes, we found that bumblebees do modify the nest by excavating outhouses, and they isolate feces in these cavities. And based on the finding in the Nome bees, this really wasn't surprising. It was mostly trying to validate that this different species also does the same thing. So then we wanted to test if this removal of feces actually helps prevent disease and prevents C. bombi specifically. So to test this, I really had to manipulate the location of poop. So we set up colonies that, in this case, they all had access to two cavities, a main nest cavity and an outhouse cavity. And we just put poop in different infected feces in different locations. So in one condition, we added infected feces to the main nest cavity. And in the second condition, we added the infected feces to the outhouse cavity. We gave them 2 weeks to live and incubate. And then at the end of that time, we dissected a sample of bees from each colony to do pathogen testing. And we also videotaped all of the colonies to count how many times the bees came into contact with the infectious materials. And so looking at the infection data, this is what we found. So this is showing infection intensity. So how many C. bombi cells we counted in each bee. You can think about this as like how infected the bee is on the y-axis. And the nest in gold and the outhouse. So colonies in which we added the feces to the nest in gold and colonies in which we added the feces to the outhouse in blue. And you can see that when we added feces to the outhouse, so those blue dots, we found no infection, not in a single bee that I screened. Well, when we added the feces to the nest location, we found relatively high rates of infection, and all of the colonies had detectable infection. When we counted how many times the bees touched the infected materials when we added the feces to each location, we also found the same trend. So when we added feces to the nest, the bees touched the infected feces the most. And when we added the feces to the outhouse, they touched the infected feces much less, significantly less. So these data suggest that waste removal and the removal of feces is the thing that prevents within colony C-bomb transmission. Having feces in the outhouse instead of in the nest significantly reduces how likely the bee's likelihood of touching and contacting infectious materials and the chance of them actually becoming infected as well. But then, does this outhouse shape itself enough to facilitate this waste removal and reduce disease? And does it improve colony fitness and colony outcomes? So this came out of this was a huge project that took a bunch of people. And it was based on preliminary data, like really suggestive data from that Simon Pania Gallego collected in Becky's lab at NC State. It was also done with a lot of help from an amazing undergraduate, Lily Blanton, who, yeah, none of this would have been possible without Lily. So yeah, just wanted to acknowledge all of the people who made this experiment possible. And so we used a 2x2 design of this experiment. So we had the outhouse condition. Half of the colonies had 2 cavities, a main nest cavity and an outhouse cavity. And half of them, we just kept them in one nest cage. Of these, we inoculated half of them at a low initial C-bomb bee prevalence. So 3 out of 100 bees inoculated with the pathogen, and half of them at a high initial prevalence. So this was 3 out of 10 of the bees inoculated. We then, throughout the 6-week-long experiment, collected a sample of 15 bees from each colony each week. We had a total of 20 colonies, and we did pathogen screening on those collected bees. We also weighed each colony each week, and then at the end of the experiment, we weighed all of the colonies again, and also counted how many bees of each caste we had in each colony to do like final reproduction estimates. We used full Bombus impatiens colonies in this experiment, and these colonies have up to 300 bees in them, so it was a lot of bees. This is what the shipment of bees looked like. So we used domestic bumblebees from a pollination company. So yeah, these bees have been bred in captivity. This is the setup in the lab. We keep them in red light because they can't fly and can be a little less aggressive if you keep them in red light. I did a lot of shopping to keep them well fed. These colonies can eat quite a lot. And just in case you haven't seen the inside of a bumblebee colony, this is what they look like. So you can see it's like chaos, like 300 bees running around. This one's trying to escape. Many of these bees escaped. We just had them flying around in the lab. It's always a good time. But so what did we find? So when we look at the low initial inoculation conditions, so the colonies in which we inoculated 3 out of 100 bees, we found no significant effect of the outhouse. So this plot is showing you time on the x-axis and infection on the y-axis. And each of those points represents one bee. So the points on top represent bees with detected infection, and the points on the bottom represent bees without any infection. And you can see we found no significant difference between that gold control and blue outhouse line. But we also just generally didn't find much transmission in any of the colonies. That line stays relatively flat throughout the whole experiment. And so it's maybe not surprising that the outhouse didn't significantly reduce infection, because it's difficult to reduce infection from no infection. And indeed, when we look at the high initial inoculation condition, we see something much different. So again, this plot is showing time, weeks on the x-axis and infection on the y-axis. And again, that blue outhouse line is really flat throughout the whole study, indicating slow pathogen transmission. But that gold control line shoots way up at about 3 weeks. And this indicates that the outhouse significantly slows pathogen transmission at a high initial inoculation condition, but not at the low initial inoculation condition. When we look at colony weight, we also see the same general effect of the outhouse. So this plot's showing you time again on the x-axis and colony weight on the y-axis. And we found that colonies without access to the outhouse, the control colonies, had significantly less colony growth and reduced final colony weight next to the colonies with the outhouse. So you can see that blue line is like way above the gold line. And if we look at reproduction, we also see the same thing. So these plots show how many adults and queens and males, reproductives, we had alive at the end of at the end of the study in each colony. And we had less in the control colonies than in the outhouse colonies in both conditions. So yes, it does look like this outhouse slows pathogen transmission and improves colony growth and fitness. But how is this happening? So the way I've set this up and what I really wanted to believe was that the outhouse slows C-bomb transmission, and that by slowing C-bomb transmission, it leads to fitness effects. You think if you have less infection, you also have improved fitness. But that's not what we found when we directly tested that. So when we plot C-bomb infection on the x-axis against colony growth and reproduction on the y-axis, we see no effect of infection on these final colony outcomes. So it seems like C-bomb isn't the thing leading to these differences in colony outcomes, which is really puzzling. And so what is causing these effects? And I have one thing I haven't told you yet, which is that we found incidental fungal infections in about 15 of these colonies, which at the time was devastating. I noticed that a bunch of my bees began dying and they had this yellow fuzz on all of them. And I didn't know why, I didn't know what it was. And we reached out to some insect pathologists on campus, Brian Lovett and Alex Lando, who absolutely saved the day. So they took these samples and identified the fungus in real time in the middle of the experiment and like really saved it. And they identified this fungus as Aspergillus flavus. So this is a widespread opportunistic generalist fungus. So it infects bees and insects, but it can also infect all kinds of different animals, including humans, which was a little bit spooky while I was doing this. It causes the disease stone brood in honeybees, which leads to the pupa being like really kind of like little stones, kind of solidified and blackened and dead. So it's quite lethal. And yeah, not good. But we found that the outhouse condition also, like having an outhouse also had a significant effect on A. flavus prevalence. So colonies with access to an outhouse space had significantly reduced A. flavus infection next to control colonies, which suggests that the outhouse not only reduced C. bombi infection, but also reduced this fungal pathogen as well. And indeed, when we plot A. flavus prevalence against the colony outcomes, we find a negative relationship. So we find that as A. flavus prevalence increases, colony growth and reproduction also decrease, suggesting that it might be at least one of the things contributing to this effect. So to sum all of this up, we found that bumblebees modify the nest by excavating outhouses in which they isolate feces. The removal of these species does effectively prevent within colony C-bomb transmission, so it has a social immunity function. And the outhouse significantly slows pathogen transmission and improves colony growth and fitness. But it's this fungus, A. flavus, and not C-bomb that might be driving these effects. And so this backs up that hypothesis that the Nome bees set into Haley and my minds, that this outhouse space facilitates and allows the bees to remove waste really effectively from the nest, and this slows the transmission of multiple different pathogens and improves colony fitness. And by improving fitness, this has the potential to lead to selection on nest modification itself, all of which could suggest that infectious disease is playing a big role in driving the evolution of animal homes and bumblebee nests specifically, which I think is so cool. This is an idea that's been around, but to date, this is the only study that's backed it up in a non-human animal species. But this also has practical, like real life applications in the ag industry. So we use domestic bumblebees in the pollination of crops, especially like greenhouse crops like tomatoes that can't be effectively pollinated by honeybees. But this comes at some costs. By breeding bumblebees in captivity, we keep them in kind of dense conditions, which can lead to a lot of pathogen transmission, which not only reduces the effectiveness of these bees at pollinating the plants that we want them to pollinate, so like those tomatoes, but it also can lead to pathogens spilling into native bee populations and negatively affecting native wild bees as well, which we, want to avoid if we can. But we found that this really simple solution of just giving bees access to a little extra space outside of the nest could effectively reduce disease, and that this could increase pollination and crop yields down the line. Um, it could also reduce pathogen spilling from managed bees into wild bees. If we reduce infection in managed bees, we also reduce those pathogens spilling into the wild bees. And in wild bees, it suggests that wild colonies may have relatively good defenses against disease, and within colony transmission may be less widespread than previously thought, which could guide subsequent study. So yeah, now I'm like really coming in on the end of all of this, and I wanted to bring it back to the beginning. So all of this began with some field observations that Haley and I made of wild Alaskan bumblebee nests, in which we found that these nests had a little bathroom space in which the bees pooped. And this seems like a whimsical, fun bee fact, and like in many ways, I think it is a whimsical, fun bee fact. But it also led to this robust set of lab experiments showing that simply by giving bees access to this outhouse space, we can improve colony fitness and improve agricultural pollination. So to me, this kind of highlights the significance of basic ecological science and natural history, not only because it reveals the hidden beauty of this planet, which I think is valuable just in itself, but also because it can reveal these unexpected and elegant solutions to actual human challenges. And I think when we lose these ecosystems and lose basic science, we also lose all of that. Who would have thought that we could simply give bees a little outhouse space to reduce disease if the Alaskan bees hadn't shown us that way? Um, so yeah, with that, I wanted to thank everyone who made all of these projects possible. The Sitnasak Native Corporation granted us access to collect some of the bees, which was amazing. None of this would have happened without that. Jay and Mary David, Josie and Sue, all in the Nome community, like granted us so much generosity and logistically and like humanly made the whole Nome project possible. Hollis Leaf, Justin, Matt, Derek, and Jessica Reichen, and Scott and my whole lab. And also funding. This was funded by NIH and the USDA. And with that, I think I can take questions if we have time.
Oh, all right.
I'm talking so fast.
That's all right. Let's see. You can maybe, we've got maybe you could put your contact information in the chat and then we can put the screen down, stop the screen sharing, and that way you'll be visible. Or maybe it's just my screen.
Yeah, I will. One sec. I'm gonna put my contact info in the chat, and then I'll do that.
Okay. And then while while Leah's putting her contact information in, this is the time of straight science when before we think of while we're thinking of our question, we throw a little love in the chat for Leah for taking the time to take time out of her evening in Juneau to let us know what you and Haley found and did with that information, which is amazing. I have much respect for both of you, and I have a lot more respect for our bees. Not only are they busy, but those guys are tidy too, and they keep a clean nest. And it sounds like with good reason, right? It helps out everything. So show a little love in the chat for Leah and Haley. Always nice to hear appreciation for taking the time to show us and educate us a little bit more about bees. I know I've learned a lot and I've got some questions. But while people are putting that in, there was a question in the chat or right off the bat. Oh, I think Haley got it about how do you separate bumblebees versus yellow jackets? Bumblebees, Haley put bumblebees are fluffy and don't have much of a waist, and yellow jackets and their relatives have a narrow waist between thorax and abdomen and no visible hair by comparison. So I hope that that helps, that answers that question.
Yes,
Fat and
that's
fluffy.
awesome.
All right. And with that, any questions right off the bat? Or you can put your question in the chat if it so be it. There's a lot of bee stuff. I was holding
I
back
know
on
so
making
many bee puns.
making puns.
And
So I know that let's see if yeah, Mary Jane is still in. One question I had, and this was also on the earlier talk, it's so fascinating because you came out here and Nome bees showed you their outhouse, honestly. And my question was, and I am completely ignorant, and Mary Jane, stand by if you're still on. Is there, or have you looked at any um, regional, local, traditional knowledge on bees or bee mythology or stories? And I say that because a lot of I deal mostly with marine mammals, kind of professional. I'm gonna do everything, but marine mammals is something trained in to me. But and there's a lot of stories that are warped and weaved into teaching moments about those animals and stuff. And I just wondered, we're so rich in cultural um, knowledge in the Seward Peninsula, Bering Strait region that I just wondered. I have no knowledge of insects, and so I was wondering if there is there any kind of older stories or I don't know who else Mary Jane would be on here that would have the knowledge. Um, do you know, is that anything you've looked at?
I don't know. So Haley and I like asked around and
Yeah,
but I was only in Nome for 3 weeks and didn't get to know a ton of people. So I definitely
sure.
did not have exhaustive knowledge. And we didn't talk to anyone who yeah, who knew. So yeah, I would love to know if anyone has that knowledge. But Haley and I didn't find it.
All right. Mary Jane, did you want to say anything on that? Because you had a story that wasn't of the region, but I had just never heard of any kind of bee stories. Um, but if you don't want, that's fine too. And if you want to unmute, it's the microphone button on the lower left, if that's where your toolbar is. But anyway, something to think about there is that expert knowledge.
Yeah.
I just wondered if someone else had stepped on the tundra and heard it buzz and had knowledge or passing that along. Haley, is that something? Is that ever you ever stumbled across anything more with that?
No, it's something I try and ask about when I can. I'm not a social scientist and definitely do not want to engage in like what would be considered social science and collecting traditional knowledge, but I'm
Mm-hmm.
like you deeply curious about stories or information that's out there. And when Leah and I were up in 2021, we had a chance to talk to Esther Burdon very briefly, and she told us a couple stories about berry picking with her sister and the bees that would fly around when they were out getting berries. But I think that's the extent um that I can remember. Definitely something that I would love to learn more about. If anybody has any connections or knows anybody that wants to talk about bees, I think that knowledge is out there. It's it's just connecting with the right people.
Right. Thank you. And Mary Jane, thank you in the chat. Mary Jane wrote, yes, I heard that if the bugs go on top of the water, that it's a warning of upcoming storms. Well, thank you for that. I'd never heard that, Mary Jane, so thank you. And there is a question in the chat from Larry Hinsman. Is there any way to treat for these diseases in cultivated or wild populations?
Yeah, so the sea bombi, the like trypanosome species, um, we don't know of a way to like eliminate it in cultivated and wild populations, but we do have some like really cool evidence that the bees kind of self-medicate themselves. Like sunflower pollen reduces infection in bumblebees. And it seems like in some cases they may selectively seek out those medicinal pollens, which is really cool. So yeah, by feeding them specific pollens, we can reduce disease, if not eliminate it completely.
They self-medicate too?
Yeah.
The
Yeah,
bees.
little chemists.
Wow.
Bees
Okay.
can
It's a very
uh,
impressive. Honestly, you see them buzzing around. First one came out in Dexter in April 21. I think I let you know. I have a new appreciation from learning from you two about the bees and they self-medicate. Honestly, amazing.
yeah.
And there's a twofer kind of question. From Sarah, and that is, do you know if the fungus is going to become more widespread and prevalent in bees with our changing climate conditions? And the second part of that question is, what is the overall population status of our bees in Nome? And so maybe hit that first one about, is the fungus going to be more widespread and prevalent?
Yeah, totally. So I think the truth is that we don't really know what's going to happen with changing climate conditions. I think we have some evidence that like some pathogens might increase, some might decrease. And this fungus specifically, I think the biggest way it could increase with changing climate conditions. So it's widespread, like it's a fungus that's in the soil all around us right now. And it only can become really infectious if it's at really high levels. And also if like the bee is kind of sick already. So one reason I think it might have been a big issue in my bees was that we gave them one pathogen and then they got hit with this fungus. But so if with changing climate conditions, that leads to increased challenges on the bees, maybe like, stress because it's too hot, stress because they don't have as much food and it's like changing, they might be extra vulnerable to that fungus that is already all around them. So yeah, I think that's what I might predict is the biggest chance of it changing with climate, but the truth is we don't really know. And a lot of bee diseases haven't been that well studied yet. So yeah, we will see, I think.
And the second question of that twofer was, what is the overall population status of bees in Nome? And maybe is there a way that if you're looking for that, if you're looking for people to help you, that might be a good thing too.
Yeah, so
How we can help you. Yeah.
yes. So it's tough to give like an overall population status summary because I think it really depends on the individual bee species. And some of those 11 bee species that we found in Nome might be doing pretty well, and some of them might not be. Um, I think generally like Bombus frigidus seems to be pretty stable, but there's also species that we found like very few of. And without like long run data, it's really difficult for biologists to be able to tell like how it's changed. So I think it's again not a satisfying response, but I think the truth is that we don't really know what the overall population status is. And in many of the species, like we just don't have the data right now. But the way to combat that is with increased monitoring, which is what the good folks at the Alaska Bee Atlas have been building on. So yeah, if you wanted to help, you could totally get involved with them as well.
Haley, can we throw your number and contact info if people are interested in the Nome
Yeah,
area to
please.
like
So
maybe they see something weird about the bees or of just a question of interest to the bees. You've got Leah's contact information in the chat. Awesome. But and
yeah.
Haley's right here. So you can hunt her down, go right into her office, that kind of
Yeah.
thing.
Yeah, you can walk right into the Coeric Tea Building and find me. I'm really excited because Leah is actually coming back to Nome in June. So if you see us out on the road system, two crazy people with nets running around, that's us. We're looking for bees, but we're also looking for nests, and we're particularly interested in nests that are maybe in town. So I've found one in a neighbor's muskox hide. Really nice, insulated, fluffy, great nest. Not great for the people who wanted to come out the hide. But we've also found a nest in insulation. So if you are hearing buzzing or maybe seeing bees come out the side of your house, definitely shoot me a message or email or anything. We'd love to come check it out. And we're really interested in how the bees in the tundra and in other potential housing options keep their nests nice and warm, even when it's cold and rainy during our summer. Because when we were out looking for the bees in the tundra, something that was really noticeable to me, in addition to the outhouse, was just how warm and dry the actual nest was compared to the cold, wet moss around it. And I really want to know how they do that. And in order to investigate that, we need to find some more nests. So if you see bees into the ground or going into the side of your house, we would love to know more about that.
Haley, what's your number? And I'll hit punch here and it'll show
Oh
up
yeah,
on there.
here. Let me type it in.
Okay, okay, great. Thank you. That's awesome. That's awesome. So we'll keep an eye, Leah. That'd be wonderful. I'm traveling a lot in June, but hopefully I can meet up with you. And if anybody in the Nome area sees these guys, by all means, give them a hand or avoid their net, one or the other. There is another, Mary Jane did put in a little bit of information there that a family of 24 up in Barrow went camping and followed a queen bee. They went back to the beehive at night and found a huge honey inside the hive and took half of it. So thank you,
Whoa.
Mary Jane, for providing that information. Really
Yeah,
appreciate that.
that's so cool. I didn't know they had such big nests.
Well, Barrow's up north. I don't know if they've got to stow it away because they have such a maybe they have a longer winter season. I don't know, something like that.
Yeah,
Very
totally.
interesting. Thank
Different
you, Mary Jane.
species have totally different lifestyles. So yeah,
So
that's
much to know. Our bees are so smart here. Imagine they must be smart
they
everywhere.
must be geniuses. Yeah.
Yeah. There's
Uh,
another question from Rose, and that says, do bees hibernate in winter and where?
Yes. So a really cool thing about the bumblebee life cycle is that it's kind of an annual life cycle. So the colony, at the end of the season in the fall, almost all of the bees die except the new queens, who've like already mated with males. So the whole colony dies and it's just those queens left, and they go and they find a nice toasty place to live. In the Nome bees, we really don't know what kinds of places those queens go. But a lot of species like pine needles, because it's kind of well insulated. And yeah, the queens just dig a hole and wait it out. So only the queens hibernate. Then in the spring, the queens come out and they find a new nest site and begin the whole colony again, which is pretty metal. So yes, the queens hibernate, but the whole colony doesn't, which is really cool.
On top of that, the queen has to do it all herself.
Is yeah, full
She
single mom.
overwinters by herself.
Yes, yeah,
Wow.
it's
And then
amazing.
starts the whole hive again?
Yep, from nothing.
That's incredible.
I
All
know.
right. Mary Jane did add when you were mentioning about where are the bees and things. She says, I've seen bees going into the nests at the other side of the Cape at Nook. So Cape Nome, east of town, about 12 miles or so. And then Nook is further east of that.
Okay.
And that's where they have the old house pits and things like that. It's a beautiful area. There is a question as well. You hanging in there, Leah? We're
Oh, I'm
we're getting
good.
our
Yeah.
money's worth out of you on this one, on this night. So thank you. And the question is from Diane, and she says, how far do bee frigidus, or bees, travel from the nest?
Yeah, that's an awesome question. I feel like a lot of bumblebee biology hangs on this question of like, what distance do the bees fly? And it's so difficult to study directly because you can't just follow a bee like you've seen them. They go fast. So in Bombus frigidus, we have no idea. But in Bombus impatiens, I have genetic data showing that they can fly up to 10 miles in one flight. So I collected bees at sites separated by about 20 miles in Ithaca, and I found bees from the same colony at those two different sites. And if the colony was halfway in the middle, they would have had to fly at least 10 miles. So these bees can probably fly a shockingly long distance. We just don't know exactly, but like maybe a few miles, maybe many miles.
Yeah, I saw actually on the slide with the Bumblebee Watch, bumblebeewatch.org.
Bumblebee Atlas. Oh, Bumblebee Watch. Yes.
Is that right? There was a dot, and I was like, what is that way out in the ocean? It's the pribilofs. So
Yeah.
they have bumblebees at the pribilofs. You know, maybe they ride in the wings of sandhill cranes. I mean, you hear kind of all sorts of stories, and maybe there's a way that the bees do it. I don't know how a bee, unless you're catch a plane, how would a bee
Like a
make it out to
wind
the
bee?
pribilofs?
I don't know. Maybe they catch some wind.
Yeah, maybe get up high and drop out when you see the pribilofs. I don't know. Very interesting. All right. With that, any other questions for Leah and Haley or Haley? We thank you so much for spending this much time answering our bee question and educating us on our neighbors that we see and we don't think about. I do really have a new appreciation. So I thank Haley and I thank you, Leah.
Yeah. No, thank all of you. It's so fun to talk about these bees.
And thank you, Mary Jane. That was a lot of good information there. Helpful. Make my mind think on these bees. And they're self-medicating. Um, and they know the plants to do that with. Think on that. Um, we'll thank you very much. And Haley and Leah, if you want to stay on for just 1 second afterwards, we'll let everyone go. Next week, Straight Science will be back on Thursday night, the 29th. And we've got Lieutenant Commander Wilkinson and with NOAA Office of Coast Survey. And she will be talking about this summer's mapping survey that will be going on in Norton Sound. And they're going to be making better charts for Norton Sound, and probably the port of Nome would be my guess as well, but we'll find out. We'll be able to ask her ourselves next Thursday, same bat time, 6:30 p.m. And with that, we look forward to seeing you next week. And thank you so much all for coming. Have a good night, everybody.